Welcome to the deep dive. Today we're getting into something absolutely essential happening nonstop inside you right now. Cellular housekeeping.
It really is fundamental. We're talking about how cells stay viable, how they manage nutrients, energy, waste, basically how they keep the lights on.
And it's not just a random jumble in there, is it? The key idea seems to be structure, compartmentalization.
Exactly. Think of it less like a soup and more like a highly organized factory with different rooms for different jobs. It's crucial.
Okay, so why? Why is that division so critical? Why not just have everything happening in one big open space?
Well, efficiency is a big part of it. You need very specific conditions for certain reactions, like lysosomes need a really low pH, maybe around 5, to work properly.
Right, you couldn't have that acidity just floating around everywhere.
Precisely. It would damage other things. Compartments protect delicate parts from, harsh chemicals or destructive enzymes. It isolates functions.
That makes sense. And this constant work,
Hmm.
it involves a huge amount of turnover, doesn't it? Things wear out.
Oh, constantly. Organelles like mitochondria, for example, might only last, maybe 1 to 10 days? So the cell is always breaking down and rebuilding. Waste management is nonstop.
So how does that breakdown work? The cell's garbage disposal system.
A key player is the lysosome. It's like the cell's recycling center or digestive fat. It's filled with about 40 different types of potent enzymes, hydrolases that break down waste.
And these enzymes, they only work in that acidic environment you mentioned.
Yeah, that low pH activates them. Safety feature, you see. If they leak out, they won't do much damage in the neutral pH of the main cell body, the cytosol.
Smart. And lysosomes handle different kinds of waste. Stuff from outside and inside.
That's right. When the cell takes in external material, maybe through endocytosis, the lysosome digests it. That's called heterophagy.
Okay.
But it also cleans up internally. Old, worn-out organelles, like those mitochondria we mentioned, they get broken down too. That's autophagy.
Autophagy. And how does the cell target its own parts for destruction?
It's pretty amazing, actually. A double membrane, often from the endoplasmic reticulum, kind of wraps around the old organelle, forming a little package called an autophagosome.
And that package then gets delivered to the lysosome.
Exactly. It fuses with the lysosome and the contents get digested. It's a really vital renewal process.
Okay, so lysosomes handle the bulk stuff, the organelles. What about smaller things like individual faulty proteins just floating around?
Ah, good question. For that, there's another system, the proteasome. These are like molecular shredders in the cytosol.
Shredders? How do they know what to shred?
Proteins targeted for destruction get tagged. Specifically, they get marked with a chain of small protein molecules called ubiquitin.
Ubiquitin, like a little destroy me label.
Sort of, yeah.
Yeah.
Multiple ubiquitins, usually. The proteasome recognizes this tag and basically unfolds and chops up the protein. It's crucial for getting rid of misfolded proteins or controlling levels of signaling molecules.
Okay, so we've got the cleanup crew sorted.
Yeah.
But all this activity, building, cleaning, it must take a ton of energy.
Absolutely, and that primarily comes from the mitochondria. They're the powerhouses, generating most of the cell's ATP through oxidative phosphorylation.
But they're not just static power plants, are they? I read they're quite dynamic.
Extremely dynamic. They're constantly undergoing fission, splitting apart, and fusion, merging together. This helps them mix contents, remove damaged parts, and adapt to the cell's energy needs. It's also a defense against damage from oxygen-free radicals, which are a byproduct of their work.
Wow. And they do more than just energy.
Oh, yes. They're central regulators of apoptosis, program cell death. They hold the triggers, essentially. And in some cells, like brown fat, they can actually generate heat directly using special uncoupling proteins.
Heat generation.
Yeah.
Instead of ATP.
Yeah, proteins like thermogenin basically short-circuit the ATP production line, releasing the energy as heat instead. Important for warmth, especially in newborns.
Fascinating. Let's switch gears slightly to the cell's boundary, the plasma membrane. It's not just a simple barrier, right?
Not at all. It's a fluid lipid bilayer, but it's highly asymmetric. Certain lipids prefer the inner side, others the outer side, and this matters.
How so?
Well, take phosphatidylserine. Normally, it's kept strictly on the inside face of the membrane.
Okay.
But if the cell is undergoing apoptosis, it flips phosphatidylserine to the outside. This acts as a powerful eat-me signal to immune cells. It's a critical flag.
A death signal, basically.
Yeah.
And you mentioned the sheer volume of traffic across this membrane.
It's immense. Endocytosis brings material in, exocytosis sends it out. A typical cell might internalize like 1 to 2% of its entire plasma membrane every minute.
Every minute. So it has to recycle that membrane incredibly quickly or it would just disappear.
Exactly. Recycling is constant and essential. This links back to the endoplasmic reticulum, particularly the rough ER, which synthesizes membrane proteins and lipids.
The RER.
Hmm.
That's also where proteins get folded, right? Quality control.
Yes, crucial quality control. It has chaperone proteins that help newly made proteins fold correctly. If they fail to fold properly, they're usually held back and targeted for degradation, often via the proteasome system we talked about.
What happens if that system gets overloaded? Too many bad proteins.
The cell triggers what's called the ER stress response, or the unfolded protein response, the UPR. It tries to cope by slowing down overall protein production and making more chaperones.
A kind of emergency brake.
Pretty much. But if the stress is too severe or prolonged and the cell can't fix the problem, the UPR will actually initiate apoptosis. It's a self-destruct mechanism to prevent harm.
Wow. So much coordination. And holding it all together, giving the cell shape and enabling movement, is the cytoskeleton, right?
Absolutely. It's the cell's internal scaffolding. Microtubules, for instance, act like highways. Motor proteins, like kinesins and dyneins, literally walk along these tracks, pulling organelles and vesicles to where they need to go.
Incredible. It really is this constant high-energy balancing act,
Yeah.
making things, moving them, breaking them down, all perfectly timed.
It all hinges on that careful organization, those compartments, specific tags like ubiquitin, and crucially, energy.
Okay, so let's bring it all together with a final thought. You mentioned energy. What happens if that just fails? If the ATP supply suddenly stops?
Well, it's catastrophic and fast. Think about all the pumps in the cell membrane constantly working, like the sodium-potassium pump, using ATP to maintain ion gradients.
Right, keeping sodium out, potassium in.
If ATP runs out, those pumps stop almost instantly. Sodium floods into the cell, water follows because of osmosis.
So the cell swells up.
Rapidly, it swells and swells until the membrane can't take the pressure anymore and it ruptures. Lysis. Game over. It just shows how utterly dependent life is on this continuous, energy-intensive housekeeping. Without it, everything falls apart in moments.
A fragile machine indeed.
Yeah.
That's all the time we have for this deep dive.